Okay, so today's session is a little bit different from the kind of SciFinder-n training that we typically do. So for today, we are focusing on the different features and search tips that you can perform on SciFinder-n to help you find information to write up different parts of your thesis or report. Okay, so we won't be going into the details of all the different functions in SciFinder-n, but we'll just focus on the different approach that you can use to help your thesis and report writing. Okay, and as always, I'll just begin with a quick introduction for the benefit of new users. Okay, so SciFinder-n is built by CAS, and CAS is a part of the American Chemical Society. Okay, so what we do at CAS is to curate scientific content, and they come from a variety of different sources, such as your peer-reviewed journals, patent documents. We also have some conference proceedings as well as dissertations. In terms of the scientific content that we cover, it's a wide range of different fields. You can see we have content that is more towards life sciences, biology, but also more towards the applied sciences, more engineering type of topics. Okay. We also got the Medline reference database in SciFinder-n. So when you search in SciFinder-n, you actually get to search the CAS reference database as well as the Medline reference database. And what is unique about SciFinder-n compared to other information sources is the indexing that we perform on the documents. Okay, so we have this team of scientists that actually reads through the full text, papers, and patents when we receive it from the respective sources. And when they are analyzing the full text document, they would extract out important bits of information, such as the key topics that are covered or discussed in your original document, any novel compounds that have been discovered or studied in the paper, or even any chemical reactions. So it's because of this indexing that we perform, we are able to then make this information discoverable and searchable via SciFinder-n. And later on, when we go through the different points of finding information for your thesis or your report, we will actually be making use a lot of this indexed information to help us locate difficult to find data points. Okay. So in SciFinder-n, again, like I mentioned, because of the different indexing that we perform, you can do a variety of different types of searches, right? It could be a substance search, it could be a keyword for references, it could be a chemical transformation search. So, lots of different search techniques to suit different types of searching needs. Okay, so as I've mentioned, today we're gonna kind of focus on how to use SciFinder-n to help us with our thesis or report writing. So what I have shown here on my slide, I believe most of you would be familiar. It's kind of like a standard structure of a report or a thesis. Okay, so very often we would start with the introduction. And introduction is where you would typically describe the state of the art of the research area that you are working on. What has already been done, right? And perhaps also highlighting challenges that are currently being faced in the area of research that you're working in. And then from there, you would kind of develop your thesis statement, because ideally, the research that you'll be doing should be typically, right, it would be trying to improve an existing problem in the area of research or solve some problems that this area of research is still facing. So in the introduction section is typically where you would do a lot of the background literature review, right? Just to kind of gather information to understand what has already been done and what else should be done or needs to be done. Okay, and then once you've set the stage with your introduction and developed your thesis statement, then it would typically be followed by the methodology section. And this is where you would kind of explain, right? What is the approach of your research or your experiment that would offer you results or help you to come to certain conclusion based on your thesis statement. So in the methodology section, you would typically be also detailing out the actual process or the actual experiments that you have performed. And again, to tackle this section, of course, if you can design an experiment or a methodology from scratch, that is an option. But a lot of times you will also be referencing on what has already been reported, right? As a guideline, or just kind of give us an idea how to approach certain experiments or methodology, okay? And then once you have outlined your methodology in your report, the next section is typically followed by the results, right? So this is really where you would be reporting on the findings of your research or experiments that you've performed. So the results section would, I won't be covering this part because this is largely dependent on what you have achieved, what is the outcome of the actual experiments you have performed for your research, okay? So after the results, then you would likely have a discussion section, right? So the discussion would be kind of trying to explain the significance of the results that you have obtained from your experiments. Whether does this support or I guess challenge your thesis statement, and also if there are any unexpected results, you probably want to highlight it as well and offer some sort of explanation to make sense of the results, okay? Again, in SciFinder-n, you could potentially use SciFinder-n to help you gain a little bit more understanding of the results that you have obtained, and also maybe learn from findings from other authors that could also give you some idea or inspiration on what could be happening in your research itself, okay? And then lastly, of course, we'll have the conclusion section. So this is, again, dependent on the outcome of your research, outcome of your experiments, and then you would then tie it back to your thesis statement to see whether, does your thesis statement, is it fulfilled, right? Or more work needs to be done, or, can you conclude that, oh, your thesis statement is not true, okay? So in terms of the SciFinder-n specs, we will be focusing on the introduction section, methodology, as well as discussion. So like I say, results and conclusion is very much dependent on the actual outcome of your research. And then at the end, I'll just kind of include this optional section where, after you've completed your research and your project, you may want to publish it. So this is just one extra little tip that you can use SciFinder-n to find appropriate journals that you may choose to submit your manuscript to. Okay. All right. So let's take a look at the first section. So the introduction section. So like I say, this is the part where you kind of want to gather information on what has already been done. So one approach to doing this is, of course, to look for review articles. So review articles are a quick way to kind of quickly learn, right? Because usually review articles would summarize what has already been done up to the stage where the review article was published. So that could be a quick way to just to get an understanding, right, of the current landscape. Another possible approach is, of course, to look at highly cited articles from your research area, because this would, it will be good to be aware of what aspects of research in the area that you're working on that has high impact or have impacted many people who are working in the same area, okay? And then I'd like to mention as well, if you look at the screenshot here, under document type, so depending on the topic that you are working in some research areas, you will actually find quite a bit of information from patent literature, which is again another advantage of SciFinder-n because you get to search both patent and non-patent literature. All right, so patent literature can also contribute to the state of the art. Okay. And then the other aspect of introduction, because like I say, you will likely be also developing on your thesis statement. So you'll likely want to then focus on the last maybe 2 to 3 years, right? The more recent publication, and just to really focus in on what has been the new up and coming things happening in your research area, okay? So for such instances, you may choose to actually want to exclude review articles because you really want to just focus on papers that are talking about new research, right? Because review articles are typically a summary of what has already been done. So in SciFinder-n, we have also the exclude function. So it's easy for you to kind of remove certain types of references that you may not want to see from your answer set. And of course, you can use the publication year filter to narrow down your references to those in the more recent years. Okay. And then lastly, again, still on introduction, depending on the situation, some of you may be given a specific topic to work on by your professor, and you can just start your kind of literature search around the area. In some cases, some of you may be given a general area and asked to develop your own focus, right? So in those cases, you not only want to kind of know what has already been done, you probably want to be able to have a good overview of what has been done more than other areas, okay? And possibly identify new or novel or niche areas for you to start your own research exploration, okay, your own R&D. So I'll be talking about the concept filter. So this is one filter in SciFinder-n that you can use to kind of have a nice overview or kind of see the trends in terms of subtopics of the research area, which subtopic has been commonly worked on, which ones less, and those that are less worked on, could it be a new opportunity for you? All right, so let's switch into the actual platform, and I will go through the steps on the different things that we have discussed so far. Okay, so we are in the SciFinder-n platform itself. First time using it, you'll be asked to log in with your username and password, all right? And this will be the landing page that you see. Okay, so I talked about finding information on the current state of the art, right? The current landscape. So in such cases, most of the time, you will likely be doing a reference search where you would use keywords, right, to help you find relevant references. So I'm going to put in my topic, which is my example that I'm going to use is aquatic weeds, and possibly to give as a source for biofuel. So you could put in just the keywords of your search this way. All right. This would also work, but of course, this is not going to be a very specific search because if you search it with just 3 words and no Boolean operators, SciFinder-n will try to find references that, of course, give you all the 3 words that you have specified, but they will also then pull in references where you get partial hit space on your query. Okay, so depending on the situation, right, both ways would be okay. But just to let you know, there is you can actually make the search more specific by for example, including Boolean operators such as this. Okay, so in this way, all your references will contain the 3 required keywords, and or not, right? All the Boolean operators you can use. In fact, you can even go more specific if you wish and put in double quotation this way so that aquatic weeds will always occur next to each other. All right. But in this case, I'm just going to leave it slightly broader. I don't want to restrict myself to only references where aquatic weeks need to occur to next to each other. I'll leave it this way. Okay, and I'm going to run the search. Okay, so because I have a relatively specific search query, I would actually go ahead and load the entire result set. Okay, if you didn't use Boolean operators, then you may want to not load the entire answer set, so you at least see the ones that are more relevant first. So this is our answer set. Now, if you look under document type, you will see the different sources, right, where the information comes from. Like I say, sometimes we may also see patent literature, which could contribute to kind of current landscape, okay, information on current landscape. And we have the document type for review papers. So this would find you the review papers. So currently my answer sets are sorted by number of times cited. I may choose to sort it by relevance first, because I really want to find review papers that are most relevant to what I have searched. Okay, so I've changed it to relevance. So you can see now the titles definitely look more relevant to our search topic. If you didn't use any Boolean operators, then I would definitely recommend you to use the relevance ranking so that references that have all the 3 hits will come up at the top of the answer set. Okay, so this is the review articles. Then if you like to focus in on the more recent papers, okay, so I'm going to just remove this. We have the publication year filter. All right, you can just slide this to maybe focus in on the ones in the last 2 to 3 years. Okay, hit apply. All right, and then of course, you know, there are still some review articles reported during this the years that we have specified. We can always go to exclude, okay, and you can exclude anything, any references based on the options that you see here. Okay, so under exclude, I'm going to choose review. So this would actually remove the 2 review articles from my answer set. So you see earlier on we were at 8. Now we're down to 6. Okay, so these 6 papers, they are not reviews, so they would be reporting on new research. Okay, and of course you may also then want to well, since it's new, it's not going to be very cited as often, but if I remove the publication year filter, okay, we can always switch it back to number of times cited, just to see which are the articles that have been highly cited by other authors. Okay, so just again, quick recap, few of the functions that you can use. Just, you know, switch about the way you sort the answers. Okay, relevance will give you answers that match your search query the best. Number of times cited for highly cited articles, and then the different document types. You can use the exclude function if you like to remove certain types of documents from your answer set. Okay, and then just to kind of let's pick an example. I'd just like to show you the indexing, right? So this has substances, so this will be a good one. So I'm just going to show you an example of the indexed information that we get from that CAS indexes from the full text. Okay, so title, abstract, okay, concepts. So these are key topics that are being discussed in the full text. So we added into SciFinder-n as well. And any key substances used, mentioned, discovered in the paper is also added in at this substances section. All right, and then you can see we not only tell you the CAS registry number, the structure, we also give you the role information to help you understand, you know, how how each of these substances are being used, right? So this has a bioindustrial manufacture preparation role, right? Whereas this, if few substances here doesn't have. So you likely can guess that there is some preparation or manufacture process maybe reported for these substances over here. Okay. All right. Now, so the other aspect that I want to talk about is the well, if we need to kind of scope a research area, right, for yourself. So let me change another topic. Let's just go to the top. All right, I'm just going to switch to another topic. I'm going to look for a fruit or a plant. Okay. So this is my topic. Okay, and I'm going to use double quotation in this case just to make it more specific. All right, so Carica papaya, and then search. Okay, so this is a much larger answer set than what we saw earlier on. And this is where using all these different filter options is even more critical, because it's not possible to go through every single results, right, in your answer set. All right, I'll still go ahead and load the entire answer set. Okay, so maybe let me change it to newest. Okay, so scoping of research area. So all these filters are great to use, but the one that is unique to SciFinder-n is the concept filter. So I've shown you the concepts, right? The key topics that are being discussed in each paper. Those concepts that you see under each reference, you can actually view all of them under the concept filter section. So this will help you to understand what are some commonly occurring concepts that appear in the references of your search. All right. So this again could also offer you a nice understanding of what are important topics in your research area. That's one thing. And then you can also use this to quickly have a quick idea what has already been done versus having to go through the entire result set. Okay, so one example, if for example, you are working on developing some new method to extract active components from that papaya plant, you probably want to know what sort of extraction techniques have already been performed, and maybe you'll like to look at those papers to see, you know, how did it work out for them, right? So I can just look for the term extraction. Okay, so you can see here, all the concepts related to the word extraction are retrieved out for you. So just by looking at this set of keywords, you kind of can get a sense what are some techniques that have been used for, you know, extraction of this sort of fruit. Of course, solvent extraction is the most common one. You see this maceration extraction, supercritical extraction, ultrasonic extraction, even microwave extraction. All right. So these are likely some newer approaches, right, to extract components from fruits. Using this, you can then say, well, if I want to maybe do a little bit more work on, say, microwave extraction, and then you can just use this to pull out, let me just click on it, to quickly look at those set of references. Okay, so this is Carica papaya, and then you can see here microwave assisted extraction. All right, and then this is also using microwave extraction, so on and so forth. Okay, so that's one way to use it. Now, again, I'll show you another example. Okay, let's, well, actually, let me just remove this filter first. So if you're working with natural products, another aspect that you could be working on is maybe trying to understand the bioactivity profile of the components that you can extract from a plant. So in this case, CES also indexed this sort of information. Just a quick little tip, if you are working with related to, you know, bioactivity, biological activity, and you want to look up concepts that are relevant to those terms, we typically name something that has a certain bioactivity as an agent of that bioactivity. All right, so like you can see here, antimicrobial agent, anti-inflammatory agent, etc. Right, so now you see all the different terms related to agent is pulled out. So you might be surprised, right? Just on this papaya plant, there's so many different types of bioactivity that people have tried to kind of study this papaya for. So for cases like this, where there's quite a long list of relevant concepts, right? In fact, there are 2 pages. It might be difficult to go through it from the table. So you can actually export this information into Excel. So what I'm going to do is just select everything here. Okay, just going to select it, hit apply. Okay, it's a total of 176 concepts that we have selected. If you scroll down all the way to the bottom, you will see that there is this option for you to download filter data from your result set. And you can actually download all of the information from this whole list of filters, not only the concept filter. Like if you want to look at publication year trends, so let me go back again, right? If you want to download this information into Excel, you can also do it, okay? Any of this information in the list, even organization, if you like to. Okay, so I'm going to go down to the bottom again. All right, I'm going to hit on this button. So you can see here, you can choose to download all the filter values or just the ones that I have selected. So in this case, I'm just going to select the applied filter values because I'm interested in just the different concepts. Okay, so I'm going to click on download. So it's downloading this file. So what happens is it will export this information into an Excel file, and it will list them according to the number of references it has been found in. Okay, so I'll just open this up and show it to you. Okay, so now at a glance, it's very easy to see, right? What is, say if you're interested in bioactivity, right? We don't talk about switching agents. If in terms of bioactivity, you can see the most commonly studied one is antibacterial, followed by antimicrobial, anti-tumor. Okay, and then you can scroll down to see which are the ones that have less references. So this could be interesting new areas for you to maybe expand the knowledge of research in this area. Okay, so you can use this as a guide to help you, select a specific research area to work on. Okay, or just to learn, what are the research trends, the trends in the research area that you are working in. Okay, so this is the concept filter. All right. So let's move on to the next part of the thesis, which is methodology. So like I mentioned, methodology is where you would list out exactly how you are carrying out the experiments or the process that you are going to be using, right, to perform the research that you have to help you answer your thesis statement. So different types of methodology information can use different approach to help you find it. The easiest one in SciFinder-n is actually reaction searching, because we have a reaction searching function in SciFinder-n that is designed specifically to find synthetic organic synthesis methodology. And in some cases, we even have the details of the methodology in SciFinder-n itself, so you don't even need to go to the full text to look for it. Okay, but that's not the only type of methodology information you can find in SciFinder-n. You can also, and I would highlight it later on in the actual examples, you can also use SciFinder-n to help you locate methodology related to, for example, analysis of compounds. Okay, it's not as direct as reaction or not as direct as synthesis, but there are ways to help you find references that report say, an analytical procedure that you may need for your methodology, okay? But of course, in these cases, you will have to then still go to the full text to look for the details. But, you know, just finding that journal, right, that may report a methodology that you can refer to can be quite time consuming unless you know the right approach to do it, right, or the techniques to help you locate it quickly. So analytical chemistry, analytical methodology, and I'll have two examples, right? I'll demonstrate analytical procedure as well as one for bioassays. Okay, so methodology. Another aspect of methodology could be to find materials that you want to use in your experiments. And this could be based on certain properties of the material. So for example, if you're working on developing certain catalysts or developing semiconductor devices or batteries, right, there might be certain criteria for the properties, for the properties of materials that you can use in the experiments. So sometimes you have no idea what materials could be suitable. In SciFinder-n, you can use physical chemical properties as a search to help you identify materials that have the suitable properties for you to use. Okay, and then the other aspect of finding materials is just, you know, finding chemical suppliers for you to purchase your reagents or materials required for your experiments. Okay, so I will focus on this few aspects. All right, finding methodology information and finding materials that you can use for your experiments. So let's go back to SciFinder-n. I'll start with the most straightforward one, which is to find a methodology for synthesis. Okay, so I'm going back to the home page. All right, so you can see here we do have a specific tab for reaction searching. So that's why I say this is actually the simplest of all the methodology searching. So I'm going to open up the drawing editor, and typically for searching reactions, you would draw out either your target molecule, or you can even draw out the actual chemical transformation that you're interested in, and SciFinder will perform the search. So in the interest of time, I would just, I'll just import, okay, something that I have already drawn. So I think it's this one. Okay. So for more details on reaction searching, you can learn this from our regular SciFinder-n trainings. So now I'll just kind of go through it a little bit quicker. So this is my target molecule. Okay, I've drawn it out, or I've imported it in, and I can be slightly more specific if I want to. And let me just bring this up, add in a reaction arrow so that you can see it's assigned as the target molecule, the product. Okay, and then I'll just run the search. So in this search, I'm actually trying to find out ways, right, to synthesize that compound. And I don't have any specific starting materials in mind. All right, so this could be something that I've designed in the lab, and then I want to actually now synthesize it. Okay, so first thing you may notice, in the first box of estrawn, it actually says 0, and then we get some hits in substructure. So this is actually again, you know, a good benefit of SciFinder, because sometimes what you're working on, right, maybe it's novel, so no one has actually tried to synthesize it. So we can't find an exact match, but we can find substructure hits, which is very similar, right? If the blue hit highlighting represents what I have drawn here. So it didn't find exactly this, but it has found something else that just have additional substituents. So the reaction conditions reported here would definitely be worth trying out, right, on your molecule as well. Okay. Then the other thing I'd like to point out, so if you notice this hit here, Okay, it's actually from patent literature. So again, just to highlight kind of the importance to also include patent literature in your literature search, because sometimes methodology information can be reported in patents. And if you're not looking at patent literature at all, you would think that no one has synthesized this. So you'll try to work out something from scratch. You end up spending a lot more time when actually someone has already done something, and this would help you to get to your goal a lot quicker. Okay. So in this case, for this specific example, if you'd like to know the specific details, because this doesn't come with the experimental details in SciFinder-n. If it's available in SciFinder-n, usually you'll see another link here. So this is not available. So in cases like this, where it's a patent, and you see that there's this patent pack icon, you can just open up the patent directly from SciFinder-n, and you'll be able to find the synthesis details. Okay, so I'm going to open up the viewer version. So in the viewer version of patent pack, you will have all the key compounds listed in the window over here. Okay, so depending on which is the target molecule that you're interested in, you can see that there are page numbers right underneath each compound. So you can actually just click on the page number. Okay, so you can see here, this brings me to this page over here. Now, if I scroll up a little bit, this is actually talking about synthesis of this compound. Okay, so it brings me directly to the page in the patent where they talk about the synthesis of a compound that I'm interested in. So I can read the details here. So instead of having to go through page by page to find the section where the experimental details are reported. Okay, so this is patent pack. Again, if it's available, ideally, if you can find the viewer option available, use it. And in this way, you can locate the compound of interest in the window here easily, okay, to find the synthetic methodology reported here. Now, so this search we have done, right, there are quite a few hits organized into schemes. So this is one scheme, one approach that can get us to this. Then you can see there are many different approaches. Now, if I scroll down, and they're all from this 2015 patent. Now, if I scroll down at one point, okay, let me just show it to you. Okay. So in this case, this scheme here, scheme 9, has the patent reference, but there's also a non-patent reference now. All right, and in actual fact, when I look into it, this is actually published by the same people that have published the patent. So this is what happens a lot of times, especially if the work came out from a company. If the work is important, right, they would definitely report it in a patent first to get protection, right, to claim that they are the first to discover this compound. So the information will be released in a patent publication first. And then a few years later, if they have time, this is what happens in my previous company was like this as well. If they have time, then they may actually write up a manuscript and then report it in a non-patent source, such as a journal. Okay, because in companies, it's a different objective, right? For them, getting things patented is more important than getting it published in journals. So what happens, you can see, is actually a 3-year lapse, right? The same information, but it appears 3 years later in non-patent literature, right? So again, this is one of the examples where including patents in your literature search is actually useful, because in cases like this, right, you actually get the information. Well, now, of course, it's 2021, it's okay. But imagine if you were searching this in 2016, right? You would think that no one has published this because this only appeared in 2018. Okay, so this is for synthesis. Now let's look at other types of methodology. So for the other types of methodology, different approach you can use. So let me go back again to the home page. The first approach is if you are trying to find, say, analytical methods for certain substances, or you have a specific analyte in mind. In those cases, you can actually start with a substance search, okay, and search for your analyte of interest. So I'm going to use a simple example, arsenic. Okay, so I want to find out how I can develop a method to analyze this from my sample, analyze arsenic from my sample. So I do a search for arsenic. So this finds arsenic as a substance. Of course, if you go into the details, the substance detail, you'll be able to find all this information, properties, spectra, etc. But what we're interested in is methodology, right? So what I'm going to do is actually click on references here because I want to find the literature that are relevant to arsenic. So I open this in the new tab. Let me just close this. So when I cross over from a substance into a reference answer set, I now have a new filter option for me to utilize, which is the substance role filter. So if you recall, in our very first example, I pointed out kind of the indexing that CAS does, right? We index the important compounds as well as how they are being used in each paper. So this is where the substance role information comes in handy, right? Let me open this up. So you can see here, there's a large set of references related to arsenic, but of course, each reference is talking about very diverse areas, ranging from things like pharmacological activity to, I don't know, biochemical processes, catalyst use, right? Arsenic is used in many different research areas. So for methodology, analytical methodology, one of the substance role that will be useful for you is the analyte role. Because if something has been analyzed, something has been indexed with an analyte role in that paper, it means that it is the subject of some analysis that is performed on it. Okay, so I'm just going to select analyte, hit apply. So this would at least reduce it from a few 100,000 to 25,000. And of course, it's still a wide variety of different analytical techniques, right? So the next thing you can you to find a suitable methodology information is to go to, let's go to, you can go to concept again. Okay, open this up and you already see one type of technique coming up, right? ICP mass spec. So if, for example, you want to do analysis of arsenic and you don't even have, like for my case, I don't even know what technique to even search for. If I look at this, it's a very good hint on, you know, what is a common technique to analyze heavy metals. Okay, we can even go one step further. So this is the two commonly used ones. Are there any other spectrometric techniques, for example? So what I can do is actually search spectrometry. Okay, so now you see here all the concepts related to the term spectrometry comes up. All right, so assume we are, you know, open, we are okay to consider any approach. Let's just select everything on this page. Okay, and then hit apply. So of course, if you have certain technique, that instrument or technique that you're looking for, you can just select that specific concept. In my case, you know, let's say we're exploring methods, we can select everything. And then you can, it's still quite a few number of references. We can still narrow it down further and see if there are any papers that have performed similar types of analysis of arsenic, maybe from similar samples or simple sources where arsenic is extracted from. So I can do a search within results. So I can use keywords to focus in the answer sets further. So maybe I'm trying to analyze arsenic content found in fish, okay? So I can maybe put in even the type of fish that I'm working with, let's say sea bass. And I put it in double quotation, so it's more specific. Okay, search. So now I only got 3 references, but this is going to be very precise, very relevant references because it has our arsenic analyte. Okay, it uses one type of spectrometry, one of these 90 that we have selected. All right, and it's they likely work with sea bass. Okay, so of course from this point onwards, you will then need to go to the full text to find the information. So if I pick let's say this example here, okay, I'm going to click on the reference detail just to show you how, you know, the filtering that we have done, how it leads to this, finding these 3 references. So of course we have sea bass highlighted in blue. Okay, concepts, it has this ICP-MS. This is also the multi-top MS, right? We don't know exactly which is the technique they use for arsenic. This will have to go to the full text to find. Other techniques, analytical techniques, they have also GC, capillary electrophoresis, gel electrophoresis, okay, many different techniques. And then here is our analyte of interest, arsenic. You can see here indexed with the role analyte. All right. So this happens to be a paper from ACS, which I have access to, so I can actually open up and we can take a look at the experimental section. So I'm going to use the full text link. Okay, so whether you can see the full text will depend if your library subscribes to that journal. So I'm using something that I have access to show you. All right, so this is the full text information. So like I say, experimental section is where we will find the methodology information. So let's take a look. I'm guessing it's probably using ICP-MS because that's a common, you know, technique for heavy metal analysis. All right, there's also here talking about metal ion extraction, which could be useful for you. Okay, so ICP-MS. All right, and then here it's where they will be talking about what is the instrument they use, what is the flow rate, so on and so forth. Okay, and then how they analyze the metal ions. And then of course, in the results and discussion, they will report on their findings. Look at the different heavy metals, and arsenic is one of it. Okay, so you can see here it also says metal ions measured by ICP-MS analysis. So you know this is the technique they have used. If you're looking to perform a similar technique, you can take a look at the conditions that they have used for their analysis. Okay, and it's, you know, also on sea bass, which is similar to, relevant to what we're working on. So this is one approach. All right, starting with a substance search. You can also start with a keyword search if you don't actually have a specific substance in mind. So what I'm going to do for my next example, okay, we're going to switch back to references, and we're going to try and find information on bioassays. All right, so let's do the topic on COVID-19. Okay, and we could be trying to develop a bioassay to test compounds for potential activity against COVID-19, right? So this is our result set, large answer set as expected. Then we can go into the concept filter. Say if we're looking to develop a cellular assay, okay? Using human cell lines. So in a lot of these cases, you may have specific cell lines that you want to use. So you want to be able to find information on how to develop an assay for COVID-19 using a cell line that you're working with. So I can actually search for things like human cell line, and we index that information as well. Okay, all the different human cell lines. So I'm just going to pick one of them. Okay, this looks like it has quite a few references. Probably it's relatively common. Okay, human cell line, hit apply. Okay, so in this set of references, you're likely going to find information, right, where they have used this human cell line, and typically it will be for development of an some anti-COVID assay, anti-SARS-CoV-2 assay. Okay, so just to quickly again show you an example, I'm going to use this for my case. Okay, so this tells me which papers I have access to, but it needs to be set up by the library first. So I'm not sure if this is set up for you. So I'll just use this as a quick way to find the reference because there's quite a few that I have access, quite a few in our answer set. So I think, yeah, okay. So this is an example. Again, I'm going to go into the full text. Okay. All right, so you can see already from the title, they are looking at anti-SARS-CoV-2 activity. For their case, they are analyzing the extract from this plant and this other compound, okay, in the human cell line. So the human cell line CALU3 that we picked is likely this epithelial cells, okay? Now, we want to find information on how did they actually develop this assay. So the same thing, we'll need to go down to the experimental section. So let's take a look. Okay, so this is where they prepare the virus. This is the in vitro, yeah, okay, so you can see here, this is information on how they seeded the cells and how they prepare the cell culture and eventually to run the assays. All right, so you can go through all this information. Okay, so these are very detailed methodology information on bioassays. Okay, so few techniques and approaches that I've shown you to help you locate references that would have detailed methodology that you can refer to for your own research. Okay. All right, and then the next part is looking for materials. All right, so this is looking for methodology. Next, we're going to look for materials. So I'm going back to the home page again. Let me just close some of this. Okay. So looking for materials, we will have to go to the substances section. And I said like looking for materials with certain properties. So if you like to search for information for substances using properties, it'll be under the advanced search field. So let me just expand the drop-down list. So you can see here, we have chemical properties, all right, biological properties, and then these electrical properties, thermal, structure related. So it depends what sort of properties you are interested in. Under experimental spectra, you can actually search by NMR chemical shifts. All right. So let's take a look at maybe glass transition temperature, which is under thermal. So this is for people working with polymers. Maybe you need to find a suitable polymer that you can fabricate for some devices that you're developing. Okay, so you have so maybe your polymer needs to be able to resist up to a certain heat. Okay, so I'm just going to use my example here, which is, let's say we require a glass transition temperature of at least 120 to 130. So I can put in a range. So these are the different formats you can enter your properties, okay? Sorry, I need to type it out. If it's a range, you need to type it out like 120 to 130, or you can use any of these formats here. So I'm just going to run a search. It's a very broad search. It's going to look for everything and anything that has a glass transition temperature of between 120 to 130. So as you can see, in terms of the hits, some are polymers, some are just normal small molecule type of answers. So if you are specifically interested only in polymers, we can go down to the substance class filter and just limit it to polymers. Okay, and then you can see some of them are copolymers, some are homopolymers with just one component. So again, depending on what is your requirement, if you don't wish to use copolymers, you only want to see this sort of homopolymers, you can go to the number of components and limit it to 1. This will get rid of any copolymers. Okay. Other filters that would be useful, for example, commercial availability, right? So ideally, you want to use a material that you can purchase, okay, because maybe your the focus of your research is not to develop a new polymer, but it's to develop a new device. So if you can use polymers that are already available, this would definitely make your research more efficient. Okay, so I've limited to commercially available polymers, and then these are some possible choices. Currently, it's being sorted by relevance. If you want to find polymers that are more commonly reported in the literature, you can sort them by number of references. Okay, I start with number of references descending. So you can see now right away the more common polymers are popping up, right? Polypropylene, polystyrene, PET, right? These are all very well-known polymers with the required glass transition temperature. If you don't want to use something that is so common, so of course you can, something that is not so commonly reported in the literature, you can switch it to number of references ascending. So these would have only one reference. Or if you like to know what are the newer materials or polymers that have entered our database, you can also sort them by CAS registry number descending. Okay, so the larger the CAS registry number, the newer it is to our database. Okay, so this is one example. Let's change our search. All right, so I'm going to the edit search and let's look at another property. Let's look at electrical properties, electric resistance, and say less than 10. Okay, I'm just putting in an arbitrary number. Search. Okay, so now we are looking for a different type of property, electric resistance. So some of them are metal containing, some are, organic compounds as well. So again, we can use the substance class. If we're looking for metals, maybe we would, actually what I'll do is I'll exclude. Okay, I'll exclude this sort of organic compounds. Okay, let's see what else can I exclude. All right, you can see that organic compound has been removed. Maybe I will exclude coordination compounds because what I'm really interested in is metals and alloys. Okay, so that compound has been removed. All right, so you can use this, use the exclude to exclude certain types of answers. So you have here different choice of alloys that have the required electric resistance that we have indicated. So I'll just choose one example to go in and again to put some perspective on how it has located this compound for you. Okay, under experimental properties, electrical, this is the property that we have selected, electric resistance. I said less than 10, right? So this falls under less than 10, okay? So if this is still, say, quite a large set of answers, we can even narrow it down to say, I want to look for metals containing certain, look for substances containing certain metals, all right? So I can do this by opening up the search within results. Okay, draw and say, I like to see materials that contain maybe iron and zinc for my choice of materials. Okay, iron, zinc, and then I'll just search it. Okay, so let's see. Zinc. Wait, I see zinc. Do I see iron? So actually, let me edit it. Okay, let's search this as drawn. I see zinc. I don't see iron. So let me, oh, okay, I'm not sure what's going on here. Okay, let me take a look. Iron, zinc. Let me try. Probably it doesn't work with two elements. Let me try with one and see if it works. Okay, let's just use one element. Let's maybe just use oxygen. Let's search it. Okay, I think my guess is it's not working because we didn't have actually a structure here to start with. So that's my suspicion. So let's modify this a little bit, okay? Just to test my theory, I'm going to put in a structure. I'm combining a structure with a property search, which is also something you can consider. So say I'm looking for oxides, okay? Different metal oxides with the electric resistance of less than 10. So this is actually also a good technique to introduce. Okay, so I have here oxygen, I have here my property, and I'm going to run the search. Okay, and I'm going to apply the same filters. And I think that should be why. Okay, let's see. So looking for different oxides with electric resistance of less than 10. It may take a while because it's a slightly more complex search where we are combining a structure search with a property. And it's quite a broad structure search as well because I've just put in oxygen. Oops, let's try it again. I think it should be able to work. Okay, maybe it's too broad. Let me put in let's put in iron and zinc because we saw some examples. Okay, so this is I've tried kind of make the search a little bit more specific so that the search time won't go too long. But as you can see here, iron and zinc. Okay, and this has iron and zinc, this has zinc, this has iron, right? So it matches our criteria. And we also got the experimental properties, the electric resistance that we have specified. Now, because what I really wanted to test was now I have a structure component in my search. If I decide to then refine it further refined by oxygen, it should work now. So let me just give it a go, okay? So I'm going to put in oxygen and I don't even need this anymore. All right, and then do a substructure search. Yeah, okay, so now it's able to find. So earlier on, it didn't work because the initial search didn't have a structure. All right, so this is also a good reminder for if you're trying to do refinement by structures over here, include a structure in your initial search. Okay, so looking for properties now, we look at glass transition temperature, and this is actually a nice example where we combine property together with certain types of materials that we like to see, right, by putting in iron and zinc right at the beginning. And then the last one is actually just searching for supplier information. So I'm switching to supplier. And this is really simple, right? I'm just going to put in my reagent that I'm looking for, say tween 20. Okay, search it. All right, so you can see here, this is listing all the different suppliers that sell this tween 20 reagent. You can find pricing information, click on the supplier name to go into the detail page to find contact information or any local distributors if available. Okay, and then of course you can always use the country filter. In this case, there's actually one from Singapore, two from Singapore. So most of the suppliers, they are from Europe, US, Japan, China. Okay, occasionally we may find Hong Kong, Singapore, some Southeast Asian suppliers. All right, so this is searching for materials. Okay, let's move on to the next section, methodology. We've covered the methodology itself, materials for the methodology. Okay, so the last part is really the discussion section. So this is slightly harder to say that you can always use this technique to find this information, because it really depends on what is the outcome of your results and what do you actually need to help you to help you explain your findings. All right. So I'm just showing you here two possible scenarios where you may require discussion to find information to help you with your discussion. So the first example here is again related to synthesis. This is in situations where perhaps you've performed a synthesis, it didn't work. Okay, and you're trying to write up in your discussion why it didn't work. So one possible approach is you could actually try to find in the literature for papers that have reported similar findings, right? They may have performed a similar reaction and it didn't work as well, and see what sort of explanation did they suggest, and could it also be the same reason in your case where it didn't work, okay? And also maybe when you're performing a synthesis, you find side products. You can also find examples in the literature where someone has performed a similar synthesis as you and see if they also uncovered side products, and that could give you some idea on what is the side product that you are observing in your reaction. Okay, so this is for synthesis. Then the other type, another scenario which will be slightly harder to, I guess, to say, just to highlight one technique that will work all the time. So this is just to maybe find information on related work, similar work, right? Not exactly the same, similar, and see if their findings can help you understand your results. So this is a situation where earlier on I talked about using Boolean operators and to make it more specific. So this is a situation where you may not want to use Boolean operators. You want SciFinder-n to help you find information, help you find references that are partial hits, because sometimes the partial hits, you're not talking about the exact same thing you're working on, but it could be similar enough for you to get some learnings from their experience. All right, so I'll just quickly demonstrate this. I think this is the last part of the thesis portion. Okay, so reaction, let's start with the reaction portion first. So I'm going into reactions again. We'll look at the side products. Okay, let me just open this up, and I'm going to again quickly just introduce the structure in so that we'll save some time. Okay. So in this case, I have actually drawn out a full reaction scheme with starting materials and product, but you can see that there are various symbols here. So this is doing like a broad search where we are just we're interested in just the actual transformation, but we're not so particular on what are the actual starting materials, as long as it's performing the same type of reaction, we are interested to find those hits, okay? Because we're trying to understand, right, side products and other aspects of this reaction. So we want to learn from what others may have possibly observed. So I'm just going to run the search, okay? So this is all good, all right? This doesn't have any side products, as we can see. So you can scroll through, right? And in some cases, if the authors observe side products, they may have isolated it and reported it. So it really depends. Sometimes the side product, it's too minor. People may not actually spend time isolating it, but we can always just screen through, right? Screen through our result set. So to see if anyone reported side product formation. So typically, if there's side product, you'll see more than one product reported here. All right, so I'll just go to page 2. Go to page 2. Okay, let's see. Page 2, page 2, page 2, page 2. Okay, I'm start seeing it. Some examples of side products. So you can see here, this plus this, and there are 2 products mentioned here. The ones that are highlighted, the product that is highlighted in blue is likely the one that we are expecting to form, okay? That matches our initial reaction search. But this paper, this author has also reported a second product. So this is an example of a side product formation. So if you happen to be working on similar, this sort of chemistry, okay, and you found that there are 2 main products for your reaction, because maybe you use slightly different reaction conditions. You got a more side product formation, and you have no idea what it could possibly be. This is where it can give you some inspiration. Okay, so someone else has seen this, right? Is it also happening for my case? Okay, so you can see here different side products isolated. Okay, this is also a slightly different side product than the first one that we've seen over, where was it? It was further down. Yeah, over here. Okay, so this is really just running a regular reaction search, maybe making it a little bit broader, right? You don't have to draw out the exact studying material that you're working with. Just make sure the reaction sites are in there. And then going through the different schemes to see if anyone reported extra products. Okay, this is one aspect. Another way you can try, so let's see if it works for this. Okay, I'm just going to open this up. Okay, no. So I didn't mention earlier, but the reaction notes is also another way filter to help you find methodology methodology information, specific types of methodology. For example, if I want to use photochemical type of reaction conditions, I can choose photochemical, or I want to use green chemistry solvent. I can use this to help me find the methodology that's using a green solvent. Okay, reaction notes filter. In some cases, reactions, we index failed reactions as well, if it's reported in the paper. Right, most authors don't report things that fail, unfortunately, right? They only report things that work. So this information is not so common, but if the author reports a failed reaction, we will actually index it as well, and it could be a learning point for you. So let me just pull out another example. Okay, and this time I'm just going to quickly draw it out. So let's see. Okay. And also just to kind of show you how actually, maybe I want to leave it a bit broad. So I'm just going to put in the oxygen. Okay, but you can see there are some pre-drawn functional groups here that you can use. Let's get this done quickly. Okay, periodic table. Okay, I'm going to do a regular kind of Suzuki coupling search. So I'm going to draw my product out. All right, search. And going to the reaction notes, I'll pick the substructure. Yeah. Go to the reaction notes like before. Reaction notes. So you can see here, these are additional reaction types, right? Microwave, irradiation, solid supported catalyst. So I'm actually looking for, all right, let me just modify this, make it even broader. Okay, I'm going to remove, I know why, because this is wrong. Okay, I'm going to the substructure again. Like I say, I want to be a little bit broader so I have more chances of finding field reactions. Okay, so you can see here different types of reaction conditions. And then in this case, like I say, this is quite hard to find. All right, but in this example, this is a field reaction that was reported and we indexed it. So you can see it's quite interesting. It actually has a reaction arrow, but there's a cross here. So this is telling us that this author tried to perform this Suzuki coupling, and there's a nitrile here. I guess they were hoping that in one step they could convert this nitrile, okay, let me just zoom in, this nitrile group into an aldehyde, but it didn't work. Okay, so if you were attempting something similar, then this is maybe a paper worth going in to look and see if they offer any additional insights on why it didn't work, and maybe they then manage to find an alternative that actually works out for them. Okay, so this is for finding field reactions. Go to reaction notes to look for it. Just remember to keep your reaction search broad, but of course the reaction sites must be intact so that it's actually still talking about the same transformation. And then go into the reaction notes to look for this reaction, field reaction index, okay, indexing. All right. And then of course, the last one we have is reference search. So let me go back again to, oh, are you kidding me? Let me just take a look. No, okay, it was a scare. All right, let's go to references. Let me type out my topic. So say I'm looking at, my topic is kind of looking at improving the shelf life of seafood, okay? So I have a specific seafood, species of seafood that I'm working on. So I have here macro, right? This is a specific type of prawns, shrimp. Okay, and if I will try to find specific information on this, right? Just trying to learn what is the current state of art on work around this, I would make this specific because I'm interested in this specific species and then shelf life. This might be the initial search that I'm doing, right? I think I got my spelling wrong, so just give me a minute. Let me figure this out. Okay. Okay, let's see. Likely my spelling is wrong. Let me just use the word suggestion to help me out. Macro. Isn't that what I typed? Let me just run this to see. Okay, it's GII. Okay, this is what. For run the search. Okay, like I say, if I was trying to find information on this for like just to understand the current state of the art, so on and so forth, I would do it a specific search, right? I put in the inverted commas because I'm looking for this species specifically, right? Looking at the improving the shelf life of these species. Now, say if I want to, maybe I encounter some problems in my research. I just want to see if people working in this area encounter similar problems. And maybe I looked through all this, I didn't find anything. Then I might actually just run a search without the double quotation. Okay. This is an example of how we can make the search broader, right? So remove the double quotation, or you can even remove the Boolean operators. So this will pull back a few more hits. Earlier on, I had 19. Now I have 23. Now, because my search is broader, right? So if I go through the answers, so these are all the exact species that I'm looking at. But you can see here now, this actually is looking at a slightly different species. It's also from the Macrobrachium genus, right? But it's a Nipposens, right? Slightly different species. So maybe, right, that paper could offer, let me see if there are any others. That paper, maybe they have some, again here, Nipposens, some other findings that could be useful for my research, all right? So this is an example of how you can broaden it. Removing double quotation, removing Boolean operators. These are some ways to broaden the search. All right. Okay, and then of course, the other aspect is, let me just kind of change it to number of times cited, is to look at cited literature as well, okay? So let me just open this up. Again, this can, sorry, what I meant to do is to open up the citation map so I can see both the citing and cited references. So this could also help you find other papers, right, that are working in the same area, but maybe not the same species, or maybe not even prawns, right, some other seafood, and see if you can learn from them as well. So this is the citation map where you can see a number of times who cited this paper, right? This is the root document, who cited this paper, and then the papers that have been cited by this root document. They are available in both tabs. Okay, you can click on the box to read the abstract. There are also filters here that you can use. If you have a long list of references, you can, maybe I'll say, let's take a look at the ones focusing on food preservation and see if I can find any interesting information from this set of references. Okay, so there are filters here for you to find more relevant cited literature. Okay, so that's that. And then the last bit, like I said, is optional, is to find suitable journals to publish in. This is relatively straightforward. So I'm just, again, using the same answer set, okay? We have here a publication name filter. So you can see for this type of research papers, what are some examples of journals that you can possibly submit your manuscript to. Okay, so just look under publication name. And then the last bit is just to mention some other useful functions that can help you in your information searching. Okay, so there is search history and calendar for you to recall searches that you have ran previously. We also have a combine function, and I'll show it to you in a minute, where you can combine results from different searches to find unique references or to find references that occur in both answer sets. Okay, so these are situations where maybe you need to do multiple searches to get to what you need. You can also create alerts, right, to keep yourself updated on any latest publication. You can also share your search results easily with your lab mates and supervisor. And of course, lastly, finally, when you're writing up your thesis or report, you need to do the citations, manage the citations of your write-up. You can export the references in citation format into whatever software you're using. Okay, so search history is here, this clock function. You can see here all the searches that we have run this morning. It's all automatically saved for us. And I can go back to rerun or edit the search. You can even go back to certain days in the month, previous months, to look up the searches. Okay, so say if I'm, just reset this, if I want to recall what I ran on the 20th of September, I just click the mouse on the end date, select 20th September, and this is what I have searched. Okay, and then you can also, okay, let me just rerun this, create alerts. Okay, so this is done by clicking on the Save button. And in fact, if you have applied filters on your search results, okay, so let's say I select food preservation as an additional filter for my result set, I can save the search and the filter applied. Okay, so in this way, I can rerun the search easily and not have to reapply all the filters. So especially if you have applied multiple filters, it will be good to save the search so that you can just rerun in one click and it will remember all the filters. And of course, when you save the search is when you can also set up your alerts. And the alerts will work based on the search as well as the filters you have applied. Okay, so just give it a name. You can choose as available, weekly or monthly, and remember to save it. All right, so let's just give it a simple name, shelf life. Just remember to save it. This will go into the star folder over here. This is where your saved searches and alerts are found. All right, this is what we have just created. You can rerun it. You can even select specific references to save together with your search, and you will see, let me see if I can find any examples. So I've not been saving specific references for a while, so let me just go to, okay, so you can see here, if I have saved specific references, there'll be a view saved button. Okay, and then the combine function is here where you can combine, say, any type of answer sets, right? Even references, even substances, reactions. You can say add, intersect, or subtract. Okay, and of course, if you like to share your search results with your lab mates or your supervisor, you can share it from here as well. Okay, I've selected this shelf life. I can click on the email icon. This will be sent from SciFinder-n. You just type in the email, or if I choose the other option, it will open up your default email app. In my case, it's Outlook. All right, so it essentially shares the link, and then you can send it to your supervisor or your lab mate. Okay. You can also share search results from this page as well. As long as you see this, icon, you can share the results. And then, of course, if you like to transfer the references into a citation management software, okay, hit on the download button and choose the citation format. Okay. Yeah, so this is all I have. Yeah, so this is all I have for my presentation. So let me see if there are any questions. Okay, so I have a question here. asking about registration again. So let's recap that because maybe some of you have missed it. We covered right at the beginning. Okay, so let me go to my browser. Okay, I've closed it. So what I did, actually, I think I searched UMT SciFinder in Malaysia. So this is your UMT library website URL. Okay, so go into it. Under e-resources, science and technology. Okay, there's SciFinder. So this will be the registration link. So if you are not on campus, I think you will need to either log on to your school's VPN or Easy Proxy. If you are not able to access the link off campus, okay? So this is the registration page. Click on it. It will ask you to accept the license agreement. All right, fill in the information here. The email address here will be the university email. Create your own username and password. Like I say, go to the tips if you need to know the rules required for the username and password. And then finally, click on register. Then there's one more step. Go to your inbox and you should have received an email from CAS when you click the register button. There's a link in the email. Click on that link. It's a confirmation link, and this will complete the registration. All right. And then to access it, you can go to SciFinder-n. I think this leads to SciFinder-n. Okay. Yeah. All right. So that's how you can register. Go to the library website and click on this link over here. Okay. Hopefully this is clear. Let's see if there are any other questions. Right, most welcome. Okay, so I don't see any other questions. So I guess the one last thing I'll just mention. This is the quiz that we are running from today to 22nd December. I will send out the recording for this session together with the link for the quiz as well. And I believe the library has also sent out this information previously. Okay, so if you're interested to take part, yeah, you have 2 weeks to do so. Like I say, it will require you to run some searches in SciFinder-n. For some of the questions, 1 or 2 questions will require you to run some searches in SciFinder-n to get the answers. So you'll need to create your account to take part. Okay. All right, that's all I have. If there are no other questions, I don't know if Sharifa, are you still around?
Yeah.
To say anything? Yeah.
Yeah. Actually, we have posted the quiz information in our portal. On our portal, so just go to the home page of our portal. Then you can find the information about the quiz on the first page of the portal.
Okay.
Yeah.
Great. Okay. Yeah. So I'll send out the recording as well when it's ready. Yeah, to all the people who have registered for the session. Okay. So if there are no further questions, we will end the session. Of course, you can always contact Sharifa or myself anytime if you have questions later on. Okay.
Okay, thank you, Pamela. And I will send the email of the recording and the do you have any the PDF file of the presentation slides?
Yeah, I can send you the PDF slides.
Okay.
Sure.
Okay, great. Thank you.
All right. Okay, thank you, everyone. Have a great day.
Thank you.
Okay, bye-bye.